Welcome back, microbiologists. We have discussed Koch a little bit already. If you remember from chapter 1, Koch developed a set of postulates. And his postulates are used to prove the cause of an infectious disease. So if you have a disease and you don't know what's causing it, you can go through these postulates to be able to prove that this is a specific organism that causes this specific disease. There are some limits to it. Before we even start talking about it, there are some limits to his postulates. One would be that some pathogens can cause several disease conditions, so it's difficult to keep up with, well, this person with an infection has these symptoms, this person with this has these symptoms. Is it actually this microbe causing these infections, or is it something else going on? So that makes it difficult to use Koch's postulates. And then some pathogens cause disease only in humans. So these postulates are going to rely on using lab animals, and we don't generally, or at least I don't, and I assume you don't, agree with using people as lab guinea pigs. So we can't go through his postulates like that. But when he showed the first disease that Koch used to show a specific infection was anthrax. He was studying anthrax to show that Bacillus anthracis was the infectious particle. Anthrax was a band in the 80s. So that if you are old enough to enjoy that. He also used his postulates to show that Mycobacteria tuberculosis is a bacteria that caused tuberculosis. So in his postulates, the first thing is that the same pathogen has to be present in every case of the disease. If half of your patients have this, with the disease, have the bacteria, and half of them don't, then Koch's postulates are not going to work for you. The second is that the pathogen has to be isolated from the diseased host and then grown in pure culture. So here's all your patients. They all have this disease. They all have anthrax. You culture this bacteria from every single one of them, and then you grow it up in pure culture. You've done your T streaks, and then you've stopped and looked under it in a microscope. You are able to study this microbe and identify this microbe. Then you're going to take this microbe and inoculate it into a healthy but susceptible lab animal. So it's healthy to begin with. It doesn't have any other infections, and it's susceptible. It's able to get sick. So you now inoculate it into your animal, and then that animal is going to get sick, develop the disease, and you are going to then be able to isolate that original bacteria from the animal and identify it that it's the same as what you started with. If at any point you do not meet one of these criteria, then you need to start over figuring out what your infectious agent is, or it's not going to be able to be used with Koch's postulates. Okay, so here you have your initial animal that is sick. You isolate the bacteria, you grow it up into pure culture, you identify it, you say, okay, this is a streptobacilli growing in this animal. Take it and inject it into a healthy but susceptible lab animal. Second little Mickey is going to get sick and have the same symptoms, have the same disease. And then you sacrifice that animal, you grow up the organism or the microbes in pure culture, identify it, and it is the same as what the first patient had. So you have the same pathogen. You're able to show that this pathogen, when inoculated in a healthy animal, causes disease. You've now been able to prove this is a causative agent of this disease. So now you should be able to explain some exceptions to Koch's postulates and be able to describe the steps involved in Koch's postulates. Okay, this is a pretty quick lecture video, but a very important one. So make sure that you're comfortable with this. Be able to describe to me, draw it out, list it out. What are Koch's postulates? If I give you a bunch of stuff, you can pick out this is the one that's not it. All right, I'll see you back for our next video. Bye, class.